The goal of this experiment is to demonstrate the feasibility to photoactivate enzyme using near-infrared irradiation. Caged protein kinase A is immobilized on the surface of upconversion nanoparticles, microinjected into cells, and kinase-induced response in living cells is observed after near-infrared irradiation.
Traditional photoactivation requires phototoxic UV light and inevitably will co-induce undesired biochemical reaction and cellular response. However, biofriendly options like near-infrared irradiation cannot trigger reaction due to its low energy.
Lanthanide-doped upconversion nanoparticle can effectively upconvert bio-friendly low-energy near-IR photon into high-energy UV photon to trigger a chemical reaction near the particle surface without direct UV irradiation.
Kinase plays a key role in oncogenesis. This technique can help us examine more signal transduction information with much lower interfering noise from UV phototoxicity and its side reaction.
Generally, individuals new to this field will encounter difficulties in modifying instruments and optical setup for upconversion application.
To begin, install a 4x objective lens next to the cuvette holder of the fluorospectrometer so that the laser can be focused in the middle of the cuvette. Place a tunable 980 nanometer laser diode 90 degrees against the objective lens.